Assalamu alaikum, dear students. We are going to discuss the life cycle of the HIV virus, and I have already shown you this. I already showed you this image in the previous lecture, just to introduce the lecture. Today, we are going to study the detail of the viral life cycle, and you have to be very attentive and try to note down any kind of new point that you're giving. I'm going to give you some short questions also on the board. So first of all, let me give you a viral overview of the viral life cycle. Then after that, we will be going over the whole life cycle. Now, what we have to do when we are writing down the question, or let me share the board with you people so that we can have some important questions on the board, and let me give you some terms also. ठीक है? तो terms जो है ना वो फिर आप लोगों को नजर आएंगी. Now we have come to the board. In the board, first of all, when you are writing the life cycle, and you have to expect a long question for the HIV life cycle in your board exam. So this is quite important. Okay? And we have many things in this life cycle which we will be discussing. First of all, you have to be very, very careful about the various parts of the HIV virus. Okay, the various parts of the HIV virus and also the functions of those parts. Okay, and also the functions of those parts of the virus, because you have to write them down. When you're going to attempt the question, first of all, you have to draw the diagram of the virus, and you have to label the diagram properly. Okay, then after drawing the diagram of the virus, you have to draw the diagram of the life cycle, because if this is a long question, then mostly when students reach the portion of the long question, they do not have much time left in the paper. So you have to draw the life cycle of the virus also. Then after drawing the life cycle of the virus, you have to give the headings of the various stages of the infection. Okay, so when we come to the headings, then we see that we have the type of receptor which is going to be compatible. That is a type of cell that the virus is going to attack. I have already told you that the virus, once it is introduced into the human bloodstream, it moves through the body, but it does not infect most of the cells, okay, that it encounters. But when the HIV virus, it encounters a certain kind of white blood cell, a certain kind of white blood cell, not all types of white blood cell, which is called the T4 cell. It begins to assault the cell by infecting the cell. Assault the cell, matlab it starts to spoil the cell. And how does it start to spoil the cell? It starts to spoil the cell by infecting the cell. Okay, now this is the type of receptor that is required for the HIV virus. The T4 cells, we all know, are the key to the human defenses against diseases. You will study this in the chapter of immunity also. Okay, they are the key cells for the human's defense system, for the जो है वो अपना क्या कहते हैं immune system के लिए बहुत ज़्यादा ये important cells हैं. And they help in the defenses against diseases. Thus, it is very, very highly significant that they are targets of HIV virus. Highly significant का मतलब ये है कि ये बहुत ज़्यादा परेशानी की बात है कि ये HIV वायरस उनको अटैक करते हैं. क्यों? बिकॉज इम्यून सिस्टम बहुत बुरे तरीके से जो है इट गेट्स डैमेज्ड. The working of the immune system gets totally destroyed because of the virus affecting the immune system. Most other animal viruses also show a are similarly narrow in their specificity, but the HIV virus is very, very narrow in specificity. When we say narrow in specificity, का मतलब ये है कि इसकी एक ही पसंद है, and that is T4 cells. T4 lymphocytes or CD4 cells. CD4 cells क्यों हम कहते हैं? Why do we call them CD4 cells? We call them CD4 cells because they have the receptors. The CD4 receptors are present on top of the T4 cells. That is why they are called the CD4 cells also. It has a special type of CD4 receptor. This CD4 receptor is actually a special type of protein which is present on top of the T4 cell. And it is through this CD4 receptor that the HIV virus holds the white blood cell and enters the cell. Like the HIV virus, we can also see some cells, some viruses that attack the human body, which are of a very, very narrow specificity. For example, poliovirus goes only to certain epithelial and nerve cells, poliovirus. We studied it in the class, and I told you people that poliovirus goes to the lower portion of the spinal cord, the nerve cells present there. Hepatitis virus goes to the liver. Rabies virus goes to the brain. Now, these are specific viruses. These have a very narrow specificity. They do not openly, they don't have a very open, wide range. Now, the next heading that we're going to see is how does the virus like HIV recognize a specific kind of target cell like the T4 cell? Now, inside our body, every kind of cell has a specific array. Specific array का मतलब ये होता है एक specific variety होता है, ठीक है? Specific array, a specific arrangement of proteins is present inside our cells. As we studied in chapter 1, के इसको हम कहते हैं surface markers. Every cell has a specific array of proteins on the cell membrane. And what are we studying? I'm now trying to tell you how the HIV virus recognizes the T4 virus in all the millions of cells that are present inside our body. The HIV virus recognizes the T4 cells because of a specific array of cell surface markers present inside them. And those cell surface markers, they are specifically designed. They are specifically designed to meet or combine with specific chemicals and specific hormones. याद रखिए सारी बातों को. HIV recognizes this T4 cell because HIV particle possesses a glycoprotein on its surface that precisely fits the specific protein CD4 on the T4 cell surface. मैंने आपको already बताया था कि glycoprotein 41 and glycoprotein 120 जो है वो HIV virus के ऊपर spikes के अंदर present हैं. These glycoprotein viruses, they match the CD4 protein on the T4 cell surface, and then it helps the T4 cell surface to enter. It helps the HIV virus to enter the T4. After fixing with the T4 cell, the HIV virus, first of all, it fixes with the T4 cell. We can say it docks with the T4 cell. Okay? After fixing or docking with the T4 cell, the HIV virus, it starts to penetrate. Penetrate ka matlab, it starts to enter. Now, these are different headings that I'm telling you. Like other animal viruses, HIV virus enters the cell by endocytosis. Endocytosis ka matlab cell eating, taking things into the cell, like we studied when we were studying the lysosomes. Endocytosis, things are going to be taken into the cell by infolding of the cell, and the viral particle will just get into the cell. Once it is inside the cell, after endocytosis, once the viral particle is inside the cell, then what is going to happen? Then it is going to start to activate its enzyme. We know that the viral particle has 3 enzymes, but those 3 enzymes are not active inside it until they get a biochemical machinery. Okay? So it is going to get active inside it. After it starts to get active inside it, what is going to happen? The first enzyme that is going to be activated is the reverse transcriptase. ठीक है, reverse transcriptase जो है, it is going to be activated. This reverse transcriptase is going to convert the RNA into the complementary viral DNA, and after that, then the attack period is going to start. This double-stranded DNA inserts itself into the chromosome of the host cell, and it becomes fixed with the host cell, and it remains in a sleeping condition, in a latent condition for many, many years, up to 8 years and 10 years. After attack, infection is not going to come forward. It is going to go through a latency period. It is going to go through a very long rest period, an average of at least 8 years. The virus genes will initiate active transcription during this period. Then after that, what is going to happen, after integrating by the help of integrase enzyme, it will start to produce its own enzymes, and then those enzymes are going to produce its own specific proteins. By the help of the protease enzyme, assemblage is going to take place. Breaking down of larger proteins is going to take place inside the human cell, in human cytoplasm. And then assemblage of viral proteins is going to take place. And the whole structure is going to be activated again. The whole structure is going to be constructed again. Now let us go back to the previous image. And in the previous image now, I will share with you people something. I will share with you people the structure of the life cycle again. Okay, let us go back to the previous image. And then after that, we will go to the detail image. Now, this is the life cycle of the virus. I hope you can all see it. I think I should share it again. Okay, now this is the first part, where the HIV, after fusion and endocytosis, it is entering the cell. And then reverse transcriptase is converting into HIV DNA. Then viral integrase is fixing it with the human DNA. And this is the latency period. And this is taking place inside the nucleus. Then HIV RNA gets activated. And the protease breaks down the proteins that are present inside the body and helps to assemble the viral RNA structure back into the HIV, and then this pinches off from the body. The next slide that I'm going to show you people is actually a detail. That is why I have hurried over this slide, because I want you people to see the details right. For the long question, you have to draw this diagram: human immunodeficiency virus life cycle. Now, this is the HIV virus, which is coming to attack the T4 cell. This is the CD4 receptor. This is the glycoprotein. It fuses with that, and then it starts to penetrate. Penetration का मतलब अंदर जाना शुरू हो जाता है. When it goes inside, it uncoats itself. It breaks off the shield. It breaks off the protein cover that is present by endocytosis. It uncoats, and after it uncoats itself, these RNA, they are going to be worked on top by the reverse transcription enzyme, reverse transcriptase, and it is going to be converted into a double-stranded DNA. This double-stranded DNA, which is the virus DNA, it is going to give us a latency period where it fixes with the human DNA. It is transported to the nucleus. In the nucleus, through the nuclear membrane, it is going to integrate. It is going to integrate, and it is going to form a provirus. A provirus means it is sleeping. It is in the form of a prophage, by the help of the enzyme, which one? Integrase. And this latency period, this rest period, it is for about 8 to 10 years. Then it gets activated. Transcription starts. Transcription means the copy of the viral DNA. The viral messenger RNA is going to be produced. That messenger RNA comes into the cytoplasm from the as the usual procedure. And then on the ribosomes, it is going to be produced. Proteins are going to be produced. Okay, and then what is going to happen after that, the messenger RNA is going to be encoded inside, it is going to be coated inside the proteins that are going to be stolen from the human body, and the amino acids that are going to be stolen and used to organize the protein. And then the coat and the shield and the capsid and everything is going to be produced, and then it pinches out of the body again. After it pinches out of the body, what happens? It takes with it a part of the cell membrane, and it totally destroys the structure of the CD4. This T4 cell. And now once this T4 cell, the structure of the T4 cell has been destroyed, the shape has been destroyed, it is of no use for the immune system. Thousands and thousands of viruses are going to be released from this. ठीक है, इसका एक दफा दोबारा से हम देख लेते हैं कि इसमें क्या होता है. नंबर वन पे है कि the HIV viruses which are responsible for AIDS, it attaches to the CD4 receptor protein on the surface of a T4 lymphocyte and enters the cell by endocytosis. ये the viral RNA is released into the cells. Yeah, it is released into the cell's cytoplasm. The DNA copy is made from the viral RNA, and that enzyme is reverse transcriptase, which is going to make the DNA copy. The DNA copy enters the chromosomal DNA of humans in the nucleus by the help of integrase enzyme. After a long period, an average of about 8 years, the virus genes initiate active transcription. ये आगे का process 8 से 10 साल बाद बनेगा. ये जो period है in which it stays inside the nucleus, it is called the latency period. ठीक है. Both the HIV RNA and HIV proteins are made. HIV RNA भी बनेगा, HIV proteins भी बनेगा, complete HIV particles are assembled. अब protease enzyme action में आ रहा है. Some cells are lysed, they are destroyed, they are burst, releasing the free HIV, whereas others bud out HIV by exocytosis. कभी कभी पूरा सेल फट जाता है, कभी कभी क्या होता है कि एक एक करके पिंच ऑफ होता है. ठीक है. Now this was all about the structure of the life cycle of HIV virus. In the next class, we will be discussing some other topic. Do join the class. I'm going to start the chapter of digestion, and then you can join me and learn something about the human digestive system in the next lecture also. Okay, see you then next time. Okay, Allah Hafiz.